Great, thank you. Cool. So thank you to the organisers for letting me contribute to this workshop. It's been really interesting and really helpful so far. So we're looking at adipose tissue biology and primarily how this is regulated genetically. And we're using Zebrafish as a model system to really look answer these questions. And we are using Zebrafish for two primary reasons. Number one is that they're transparent, and as Steve Farber and John said, that you can incubate the fish in these nice kind of fluorescent lipophilic dyes, and you can very nicely see the lipid that's accumulating in live animals. So this is a Nile red stain here, which shows neutral lipid in yellow. And the second reason is that they're amenable to kind of genetic and chemical screens. So it's a good tractable model for kind of probing the genetic basis for kind of various adiposity traits. And so I'm a developmental biologist by training, and what I'm going to talk to you today is kind of about the development of adipose tissue. So during these kind of fast growing rates that we've kind of heard about, so in juveniles or post-embryonic larvae, generally around between 1 and 2 months of age. Really the take home is that zebrafish adipose develops in different stages, and diet affects adipose development in a kind of stage-specific manner. So potentially very important on adipose development. So I think, there we go. So just to give a brief intro on adipose tissue, I'm sure we're all aware, but adipose tissue is this kind of distinct tissue, this distinct organ distributed throughout the body and accumulates lipid. So you can see here in this DEXA scan of a human, lipid is in this kind of yellow color here, and it's in all of these kind of anatomically diverse places. And so structurally, adipose tissue is formed primarily by fat cells, also called adipocytes, which are these large spherical cells shown here in green that kind of inflate with lipid and can grow to these very large cell diameters. So you can see that this cell here is possibly 100, 150 microns in diameter. And so these incredibly morphologically unique cells. And these cluster together to form an adipose tissue. And you can see here, these are both in zebrafish. You can see that there's a big adipose tissue actually associated with the pancreas in the zebrafish. All right, so medically we're interested in adipose tissue because of obesity and body fat distribution. And these are two things that we're really looking at in zebrafish. Okay, just to give you a little bit of background on what's known about zebrafish adipose tissue. It's a very small field, and actually there's very few papers on the subject. But one of the first papers was done by John, who showed that zebrafish adipose tissue is morphologically, molecularly, and actually functionally very similar to mammalian white adipose tissue. That is, it accumulates lipid in times of energy excess, and it mobilizes lipid during times of energy or caloric depletion, energy depletion. So morphologically, zebrafish adipocytes, fat cells, are labeled here with the asterisks, and you can see that they're huge. And they seem to express similar markers to mammalian adipocytes, so things like PPAR gamma and FABP11, which are kind of very established markers of mammalian white adipocytes. And so these are the same animal here in the kind of vertical columns. When they're live, they've been stained using Nile red, so you can see the lipid stored in lipid droplets, and then they're fixed and processed to look at PPAR gamma, mRNA, and you can kind of track where the cells are with the lipid expressing, say, PPAR gamma or FABP11A. So they look as though these zebrafish dipocytes look as though they're, morphologically and molecularly similar to mammalian white adipose. And this is really, one of the reasons why diet is really important, for our research. And that is that zebrafish adipose tissue doesn't really appear until, post-embryonic stages, so 10 days post-fertilization or 8 days post-fertilization. And so this means that, experimentally, when we look at days, traditionally we'll look at days post-fertilization in zebrafish, but that's not really sufficient to explain a lot of the variability that we see with adipose tissue. So again, these are nice Nile red images from John's lab, showing that initially at 5 days post-fertilization, you see lipid in the intestine and no real kind of lipid stored in the adipocytes. But then at this post-embryonic stage, so that these guys are getting fed from 5 days post-fertilization, but then shortly after that, or a good few days after that, they start to store lipid in the adipocytes, and then this kind of expands. And so a different study, another study from Kirsten Sadler's lab, basically showed that, again, if you use DPF as a metric to study adipose tissue in zebrafish, there's huge variability. So you can see that at 12 DPF, fish of this length, there's a huge variation in size of the fish for starters, but only a few of them have adipocytes. So there's huge variability. And this is primarily because the fish are independently feeding, and they're growing at vastly different rates. So Dave Perishy did a study where he looked at the relationship between fish size and fish development during these post-embryonic stages, and he showed that a fish that were 20 days old, shown here in black dots, covered a huge size range. So there were massively different sizes, ranging from 5 millimeters in length to 10 millimeters in length, and they also kind of encompass this huge developmental range as well. So you can see here, these are kind of post-embryonic developmental stages, these kind of DR, AR stages. And then you can see here, like, even within that system, there's huge variability as well. So a fish that's 6.3 millimeters could be any one of these developmental stages here. So it's really important, and I think that this needs to be incorporated into any kind of evaluation of dietary success. Is using this established kind of staging criteria that was established a few years ago by the Perishy lab. And then diet and other kind of environmental factors can affect this interaction between size of the fish and developmental stage of the fish. So for two different traits, both melanophore development and also the appearance of adipose tissue, fish that are fed, that are grown at different densities, so low density and high density, and that's the same for this experiment here, as a proxy for kind of food per capita, so food availability. So obviously low density, fish grown at low density have more food available to them and grow at a faster rate. What can be seen for both of these traits is that fish with which are grown at a high density, that have like a low food availability, actually develop or developmental traits appear at smaller sizes. So there's a disconnect between size of the fish and kind of developmental progress of the fish. And so this makes for extremely complicated comparisons or to be made at these stages. and should definitely be included in any kind of future kind of diet study. So just to kind of concentrate a little bit more on adipose tissue, so we kind of carefully characterize where adipose tissue is stored in zebrafish. And there's lots of different anatomical sites of adipose accumulation in zebrafish, but they can be broadly classified into internal adipose tissues or subcutaneous adipose tissues. So just like in humans or mouse, essentially. And the internal adipose tissues are primarily composed of visceral adipose tissues associated with the internal visceral organs. And we see very interesting dynamics in how these adipose tissues grow. So these are, so on the x-axis are the size of the fish, and then on the y-axis for this upper graph is the kind of growth in body area of the fish. And this is about 300, 400 fish here. So this is kind of a line plotted to show the growth. See, there's very consistent growth in body area across these kind of fish sizes. But if we look at the growth of the different adipose tissues, you can see that visceral adipose tissue undergoes this, appears first, but undergoes this very quick expansion that slows and plateaus out to essentially not growing at all. And then kind of opposite to that, or complementary to that, subcutaneous adipose tissue appears slightly later, but then kind of grow as visceral adipose tissue is slow, the growth of visceral adipose is slowing, subcutaneous adipose tissue is kind of expanding at a very fast rate and quickly catching up to that. Okay, so and if you look at, so these are the same graphs that I've just shown, but if you look at body fat percentage across the different sizes of fish, you can see that there's, during this kind of early phase when visceral adipose tissue is expanding really quickly, there's a very fast rate of body fat accumulation. And then this seems to slow when when lipid storage changes to subcutaneous adipose tissue, this kind of rate of body fat accumulation slows. So there seem to be two different kind of growth phases of adipose tissue in zebrafish. The first one, which is before around about 8 millimeters, is kind of characterized by very fast visceral adipose tissue growth and a very high rate of fat deposition. But then the later stage of adipose growth in zebrafish is characterized by this kind of plateauing of visceral adipose tissue growth and kind of lipids starting to be stored in subcutaneous adipose tissue, but at a lower rate. Okay, so what I'll talk about now is how a high-fat diet affects adipose growth in these two different stages. So, as mentioned by both Steve Farber and by John, we use this chicken egg yolk as kind of like a crude high-fat diet, obviously a very lipid-rich diet. And experimentally, the fish are fed our normal kind of regimen, but every day they're immersed in this 5% chicken egg yolk, almost as a supplement, to kind of give them a lipid load, essentially. So you can see that we'll raise our fish normally until a certain kind of developmental stage, and then we'll split the siblings into either a group that's immersed in system water, so a standard fed group, or a group that's immersed in system water plus the high fat diet. And then we do that typically for 1 to 3 days, or 7 days, or 2 weeks, or 3 weeks. Those are typically the kind of durations that we kind of immerse them in the high fat diet. And you can see that the effect of this high fat diet doesn't really have an effect on somatic growth. So the fish don't grow larger, but there's a small bump in adiposity. So it basically increases, it produces a response in the adipose tissue and leads to increased adipose storage. In no way does this resemble obesity, but it's a very early kind of marker of adipose remodeling in response to a high-fat diet. And really the take-home is that if we do that during this early kind of visceral-centric growth, we get, all of the high-fat induced growth occurs in internal and visceral adipose tissues, and there's almost no response in subcutaneous adipose tissues. Whereas if we do it in later stages, when subcutaneous adipose tissue is the primary type of adipose tissue that's growing, we see a response in subcutaneous fat and not visceral fat. So it seems that the different stages, if you give the fish a high-fat diet, at these different stages, different adipose tissues respond in a different way. And so that's a high-fat diet, but we've also done a similar thing with starvation, and then looking at how adipose tissues redeposit lipid in response to refeeding. And we see a similar thing in that during, and this is an example of starvation and refeeding during the later stage, the kind of subcutaneous adipose growth stage. And we see that when you starve fish, so you can see here they're losing lipid, and then when you start to refeed the fish, they're regaining lipid. And if we split this out into internal and subcutaneous adipose tissues, the colored bars represent the size and stage batched normal kind of expected levels of lipid. You can see that the internal adipose tissues have lost the ability to recover to their normal kind of fed size, whereas the subcutaneous adipose tissues kind of respond really nicely. And this leads to changes in body fat distribution. So, so basically, I guess what I'd like to kind of emphasize is that zebrafish, adipose develops in these two different phases, and if you manipulate the diet during these distinct phases, you get different responses. And so I'll just leave it at that. And a lot of this work was done in John's lab. Okay, thank you.
Is there a thought or evidence that visceral fat, it's still thought that in like people, their belly fat is more contributing to syndrome X or it's
the bad fat.
Bad fat, yeah.
Yeah.
Even if it's all white fat, but it's still location.
I mean, so I think it's interesting. So we don't have a lot of data or real kind of causative, like strong data that the visceral fat is causing a lot of metabolic kind of dysregulation. But I think it's interesting that zebrafish initially store fat around the visceral organs. And we're probably all aware that when you start feeding the fish at kind of 5 days, it takes them maybe a week for a week they're very vulnerable, I think. And then, and basically as soon as they start getting more stronger and start growing really well, they're storing fat around the visceral organs. So I think that might, it might signify a beneficial effect for the fat.
Is there any evidence for relationship between adipose tissue and egg-associated inflammation?
Yeah, so I haven't shown it, but if you put these fish on a high-fat diet, then there's increased inflammation in the adipose tissue. Yeah, so and you see that very early on, from like maybe 3 days after they've been in a high-fat diet, which is something that is occurs in mammalian, mouse and human obesity, as you get this kind of influx of immune cells. So it's definitely something that we're interested in.
I have one more question. So the demarcation which you offer, kind of 8 millimeters plus minus.
Yeah.
Is that? Do you have ideas on what anatomically or physiologically you separating that? And then I guess I would offer that up to the group also. Like, are there other lipids that are happening in the animal around that stage that may explain why they're being shifted.
Yeah, so I mean, I really don't know why there's a shift from visceral to sub-Q, and why there does seem to be probably quite a wide window that it takes to fully for subcutaneous fat to fully catch up and undergo this kind of exponential expansion.
That's interesting because total yolk sac absorption at 28 is around 7 days.
Yeah.
This is much later, right?
So you said what day was it?
We typically do. Well, this is probably around a month of age.
Oh,
But maybe.
8 millimeters.
Yeah. But I don't know why. My feelings are that the visceral fat has a beneficial role, for organ function early on, and then once the fish is through this kind of dicey phase and they're growing nicely and they're getting overfed on these zebrafish diets that we feed them, they have all this excess energy that then gets kind of redeposited into subcutaneous fat. But I don't, that's all hand-waving.